Thank you so much, Alan. It's a pleasure to be here, and I thank you for the opportunity to present on behalf of Adverum. As we heard earlier, there's still significant unmet medical need for patients with wet AMD and their providers. Topping that list is a need for more durable treatment options that improve and preserve vision. Ixo-vec is an intravitreal gene therapy that utilizes the 7-amide capsid that was created via directed evolution to effectively cross the ILM and enable transduction of retina and continuous intraocular aflibercept production. We have demonstrated durable aqueous levels that are in the therapeutic concentrations in patients with wet AMD for 5 years after a single intravitreal injection. The Ixo-vec clinical program was initiated in 2018 with the first in-human OPTIC study in which we evaluated the 2 higher doses of 6E11 and 2E11 combined with relatively short courses of steroid prophylaxis. I'll briefly walk you through the 4-year results, focusing on the 2E11 dose cohorts as this dose was advanced to the phase 2 program. In OPTIC, patients had received on average 10 annualized injections prior to receiving Ixo-vec in the trial. As you can see, over 50% of patients remain injection-free year over year through 4 years after the single injection. This was accompanied by maintenance of vision and fluid control throughout the study period. Ixo-vec was generally well tolerated. Inflammation, when present, was responsive to locally administered steroids and resolved, 100% of cases resolved by the 1-year time point. In the phase 2 LUNA trial, we evaluated enhanced corticosteroid prophylaxis regimens, both locally administered and systemic, in combination with Ixo-vec at 2E11 and a new lower dose of 6E10, with the objective of identifying the phase 3 dose and prophy regimen. Consistent with our observations in OPTIC, you can see significant treatment burden reduction, again, in patients that entered the study having received an average 10 annualized injection prior to being treated with Ixo-vec. And this was regardless of dose. Both doses demonstrated this significant treatment burden reduction, with the 6E10 dose achieving 54% freedom from injection at 52 weeks. When we look at a subgroup of patients that had received only up to 6 injections the year before, 75% of these patients were free of injections in 52 weeks. And again, this was accompanied by maintenance of BCVA and fluid control with both doses. Importantly, in LUNA, we observed greater fluid control in patients that entered the study with CST greater than 300, and this was regardless of the need for supplemental injection. Ixo-vec had a favorable safety profile and was well tolerated in LUNA. No Ixo-vec-related SAEs were reported. None of the bad stuff that Dhaval was also mentioning were reported in this trial. Local corticosteroids alone effectively minimized inflammation at both doses, and we have since selected the 6E10 dose plus difluprednate to advance to phase 3. Importantly, we heard from patients that overwhelmingly they preferred Ixo-vec to their prior treatments, would treat their second eye if they had bilateral disease, and would recommend it to their family and friends, and this was a pre-specified patient preference survey. 100% of patients with Ixo-vec, 6E10, and difluprednate responded yes to these questions. We have 5+ years of clinical experience now that has really led to an optimized product profile with Ixo-vec at 6E10, with enhanced safety, and truly the best option to provide injection freedom for patients over the long term. This is the phase 3 Artemis trial, which we initiated earlier this year. It's a non-inferiority design study with the objective of evaluating whether Ixo-vec 6E10, a single injection, will achieve comparable visual outcomes to on-label aflibercept at 2 milligrams in a broad patient population, both treatment naive and previously treated. I hope that you agree with me that these data are exciting, and we are aiming to really meet the unmet need for these patients and providers. And these data would not have been possible without, obviously, our patients who participated in Luna and OPTIC, their caregivers, our esteemed investigators, and their study teams. Thank you so much.
Congratulations. I mean, again, another story of resilience and persistence that the team has really put forward to bring forward. So you mentioned you're doing a treatment naive and previously treated population. As a parallel, would you consider just doing a treatment naive study as well?
Yeah, excellent question. So we think really, as Peter mentioned in his panel discussion, the ideal patient population for gene therapy is really broad. And so we think that having treatment experienced patients without limitations as to how experienced they are, which is the design of this study, is going to provide the confidence that retina specialists need in order to be able to make informed decisions about their patients. And so we think that both studies would be conducted in mixed population and have agreement from the FDA on this phase 3 design. So it's de-risked also from a regulatory perspective.
Thank you.
I like the flat visual acuity in injection patients. We see a rise in treatment naive visual acuity and then plateau and then plummet. So
that's right.
This is what I'm hoping we get. First in human gene therapy approved in the subretinal space, 2017. And now 3 gene therapy programs, 2 in the office, 1 in the OR, and actually in the office too. And maybe we'll see something 10 years and we'll bring it back to retina. We'll see. Thank you very much.
Thank you.